am Juan Diego Gomez.
And I am Sara Gomez.
And today we're going to talk about an article called Development Consequences of the ATG7 mediated autophagy in humans.
Autophagy is a cell mechanism to protect ill cells from pathogens, from damaged organelles, and protein aggregates, and it helps to recycle metabolites. First, we have an autophagy stimulus, that is the signal that is received by the ULK1 complex, and it helps to form a double bound structure called phagophore. The phagophore needs to expand, and to the expansion we need an interaction between many proteins that are ATG proteins, and this conjugating process is mediated by the ATG7. It is like the director that helps the conjugation. This helps to form the autophagosome. Then inside of the autophagosome we have the cargo, that is the organelle or protein we have to eliminate. So the LC32 complex linked to the organelle to the cargo, it can link directly or by a receptor, that is the P62. Then the autophagosome fuses with the lysosome, and the lysosome has electrolytic enzymes, so it helps to degrade the cargo.
So, ATG7 encodes an E1-like enzyme that activates ATG12 to ATG5, promoting the expansion of the pre-autophagosomal phagophore. ATG7 also facilitates the lipidation of the protein LC31 to generate LC32.
General objective. The objective of this investigation is to highlight the importance of ATG7 gene in the autophagy process by the study of 5 unrelated cases of recessive ATG7 variants that were damaging because they affected the function of the proteins. It's important to note that these 5 patients had a neurodevelopmental syndrome.
We will now discuss the methods used in this research, the first being immunohistochemistry. This is the identification of a tissue by a specific interaction between an antibody and an antigen. The antibody is labeled and the cell is stained to demonstrate the presence and localization of a molecule of interest. So, the sequencing methodology. It is divided into methods that have some aspects in common, like the use of radioactive and chemical methods, electrophoretic tools, and the use of original fragments from the DNA. In the chemical method, we found a based chemical hydrolysis and an original design applicable to DNA sequence with less than 200 fixed nucleotides. This method can be described in 3 phases: DNA labeling, chemical hydrolysis, and analysis of the products. In the enzymatic method, we found that the DNA is not degraded, but the synthesis of a complementary strand is interrupted in a controlled manner. This process is described in 3 sections: enzymatic analysis, fragment analysis, and automation.
Immunoplot is a laboratory technique used to identify specific proteins present in a tissue or in a cell. It is useful for diagnosis because it is affordable and effective. So, what is the process? First, we put the proteins in an electrophoresis cell charged negative, and they're gonna be separated by their sizes. In second place, we put these proteins in a nitrocellulose membrane, and then we incubate with antibodies directed specifically to the antigens present in the protein. They're going to be incubated, and there's going to be a reaction between the antibody and the antigen, and we're going to identify them by some techniques, and we're going to see the protein which the antibody was directed to. Immunofluorescence. It is a laboratory technique that uses fluorophores to visualize specific antigens present in a cell. And visualize is localization. So it consists of incubating the tissue or the cell with a specific antibody directed to the antigens. And this antibody is linked to a fluorophore. A fluorophore is a substance that emits light. Then we're going to wash and if there was a reaction between the antibody and the antigen present in the sample, we are going to watch the results in a fluorescent microscope.
Now we're going to talk about the results of the whole investigation. The following panel is a complete with a general account the results of the 5 families members affected by ataxia or ATG7 variants. We found 4 forms: circles that mean female members, squares that mean male members, diamonds unknown members, and shaded symbol affected members. Under the figures, we found the ATG7 variants that affect the family members. We conclude that the family members affected by ATG7 variants, we're gonna suffer the above mentioned alterations. Now, the monoblood analysis showed the following results. In the biopsy indicate that ATG7 was undetectable. ATG7 levels were severely decreased in the patient myoblast, and the P62 high levels were found in the patient's myoblast.
It's important here to see the ATG7 and the P62. In ATG7, we can see that in patient 1 is not present, but from patients P3 to P10, it is reduced. And it is important and interesting to notice that P62 is inversely proportional to ATG7, because we can see that, for example, in P1, in patient 1, there is more P62. So the less ATG7, the more P62. But why? It is because we saw that P62 is the receptor for the LC32 molecule that recurs the cytoplasmatic cargo. But ATG7 is important for its lipidation. So if we don't have ATG7, we don't have LC32 functional. So we're going to have more P62 free. In this analysis, they was added CQ, that is chloroquine, and AZD8055, that is a substance that is an inhibitor of mTOR. mTOR is like a protein present in the cell that contributes to cell proliferation. So adding this, cell proliferation is inhibited. So it's like to stimulate autophagy. Chloroquine is the opposite. It inhibits autophagy. So here we have that even after adding an autophagy inducer, in, for example, family 1, patient 1, we have few LC31 and few LC32. In this analysis, there was an introduction of plasmids encoding wild-type ATG7. It means that is a functional ATG7, and we compared the wild-type and the no plasmid. So we can see here a difference. We can see that ATG7 is present in the wild type. Here we can see a densitometric analysis of the ratio LC32 GADPH, that is the control, normalized to wild type values. It means it is the ratio in wild type values. So here we can see that wild type is 100% and the rest of all are the variants.
This whole clinical research made clear the importance of autophagy in physical and mental integrity of humans. This investigation strengthens our understanding of autophagy and its influence in disease and genetic significance.
Finally, we have the conclusions. So first, we know that autophagy is an essential process to keep the cell protected from cytotoxicity, and several genes are implied in the process. Molecular biology helps us study processes that are not available to our eyes or even through a microscope, know which proteins are implied in the genes that qualifies for its synthesis. Second, the molecular techniques are fundamental for the analysis of genetical diseases and the diagnosis, because it helps us knowing which protein is involved immunopathology by techniques such as immunoblogging. And finally, we conclude that molecular techniques give us a light of how to treat genetic diseases so damaging as the one in the present study, by methods such as genetic therapy.
Gracias por su atención.